In this lesson, we're going to learn about the physiology of the nephron. Now recall from the previous lesson that we named the different segments of the nephron. We have the afferent and efferent arterioles, the glomerulus in between them, and then blood is pressure filtered at the glomerulus so that filtrate comes out here into Bowman's capsule and then travels through the proximal convoluted tubule, the descending and ascending limbs of the loop of Henle, the distal convoluted tubule, and the collecting duct. And now we're going to look at what happens in each of these places. Also recall that the nephron is one of about 1 million in each kidney, and that it sits both in the cortex, the outer part of the kidney, and in the medulla, the deeper part. And that boundary is right about here. So this boundary that we're going to draw here is between the cortex of the kidney and the medulla. And we said that at the glomerulus the blood is pressure filtered. So if we look at 100% of the fluid coming in here to the glomerulus in the afferent arteriole, only about 80% of that fluid is going to leave in the efferent arteriole. 20% of the fluid that comes into the glomerulus is getting filtered here into Bowman's capsule. And the fluid part of the blood is called the plasma. So 20% of the blood plasma is filtered. Now, that's just the fluid parts with the dissolved solutes. Typically, under healthy conditions, there are no cells that make it through in that filtration, and there are no large proteins. Those get caught in the filtration process and continue out through the efferent arteriole. And there are cells wrapped around the capillaries here that form a kind of sieve or colander that can open and close a little bit to let more or less fluid filter out here. So again, fluid is filtering from the glomerulus into Bowman's capsule. And those cells that can open and close the filtration slits are called podocytes. Pod for foot and cytes for cell. They have these foot processes that stick out like this, and if this is one cell, then there's another cell like this, and they're almost like interlocked fingers that can separate out like that to leave more of a gap for fluid to pass through, or close back up to leave less of a gap. So the podocytes open and close here at the glomerulus, and the plasma is filtered and then moves into the proximal convoluted tubule. Just a note on terms here, I've got a list of terms at the bottom. Filtration is what is happening at the glomerulus, and it's that pressure filtration that we're talking about. When we reach the proximal convoluted tubule, we have reabsorption and secretion. Reabsorption means that we are moving solutes from the filtrate, which is on its way out of the body, because this is going to pass to the bladder, the ureters, and out of the body. Reabsorption means moving something from the filtrate out into the capillaries around it. So again, remember that there are capillaries around here. Those are called peritubular capillaries, as we mentioned in the previous video lesson, and there are capillaries down here in the vasa recta. They're just not shown in the diagram. So the things that we are moving out of the filtrate are going to end up in those capillaries and stay in the blood. And so that can be here things like sodium and water and nutrients like glucose. Those are all reabsorbed at the proximal convoluted tubule. Now just a note about the kinds of concentrations and volumes that we're talking about. It turns out that about 180 liters of fluid is filtered by all of your glomeruli in your kidneys every day. So 180 liters of fluid gets filtered out of your blood and is on its way out of the body. Now, if you think about it, you only have about 5 liters of blood altogether. So that's about 36 times your total blood volume, and you certainly aren't urinating out 180 liters a day. So what's happening to all of that fluid? Well, it's being filtered here, but the vast majority of it is being reabsorbed, and a lot of that is reabsorbed here at the proximal convoluted tubule, gets back into the blood, and carries on. Now just a note also about concentrations. The concentration of solutes in the blood is about 300 milliosmoles per liter, or 300 milliosmolar. And that's the concentration of the blood in the capillaries in the glomerulus, and that's not changed when that blood is filtered. So the filtrate here has a concentration of about 300 milliosmolar as well. The proximal convoluted tubule also doesn't change that. When it reabsorbs stuff, it's reabsorbing isoosmotic fluid. So it's not changing the osmolarity of what's going through because it's reabsorbing the right concentrations of solutes and water to keep this about 300 milliosmol still. The sodium is reabsorbed, for example, by active transport, and water is reabsorbed by osmosis. There's also secretion here at the proximal convoluted tubule. Remember, reabsorption is taking stuff from the filtrate back into the blood. Secretion is going in the opposite direction, and there's secretion of things like hydrogen ions here. So now we have a filtrate that has had a lot of the fluid removed and a lot of solutes removed, so it is still at the same osmolarity. It's about 300 milliosmolar as it is coming down here. I'm not going to write milliosmolar every time, but that's the units that I'm using. And then we reach the descending loop of Henle. And the descending loop of Henle is permeable to water, but it is not permeable to sodium. And so water is reabsorbed by osmosis. So water is leaving here by osmosis. And if you take a look at these concentrations in the medulla, because we've moved now from the cortex where the glomerulus is down into the medulla where the loop of Henle and the collecting duct are, if you look here at these concentrations, you can see that the medulla is getting more concentrated with depth. The osmolarity of the medulla increases with depth. And so even though water is being reabsorbed up here, it can still be reabsorbed more down here because the filtrate is entering an environment that's surrounded by an even more concentrated interstitial fluid around it. And so water just keeps leaving here, and that brings the concentration in the filtrate in the descending loop of Henle all the way down to about 1,200 milliosmolar here at the bottom, as water has continued to leave by osmosis. And then the reverse happens on the way up. The ascending limb of the loop of Henle is permeable to sodium, but not to water. And sodium is first moved by diffusion, but then after that, when necessary, it's moved by active transport. And so sodium is being pumped, that'll be my pump, sodium is being pumped out here in the ascending limb of the loop of Henle. And that active transport, coupled with the sort of bended setup here of the loop of Henle, is what actually allows this concentration gradient to be set up in the first place. Now, we said that this osmosis of water in the descending limb of the loop of Henle has resulted in a really concentrated filtrate down here at 1,200 milliosmolar, but now we've pumped a bunch of sodium out, and so that's been diluting this filtrate. And as we get up here towards the top, as we cross back from the medulla into the cortex, it's actually really quite dilute. And it can be as dilute once it gets up here as about 100 milliosmolar. So this is a very dilute filtrate. And if this were to continue passing through all the way out the body, then that would be a very dilute urine. And as we'll see in a moment and go into more detail about in the next video lesson, that's going to be really important for the function of the collecting duct in allowing us to produce either a really dilute urine when we are overhydrated or very well hydrated, and a very concentrated urine when we're dehydrated and need to conserve water. But first, let's take a look lastly at the distal convoluted tubule and the collecting duct briefly. In the distal convoluted tubule, there is selective reabsorption and secretion. So in the proximal convoluted tubule, there had been bulk reabsorption. Now it's more selective. So again, we can reabsorb sodium and glucose, and hydrogen ions might be secreted, and this is selective reabsorption and secretion. The proximal convoluted tubule takes care of getting the volume down from 180 liters to something much smaller, and the distal convoluted tubule is doing sort of the fine-tuning at this stage. Again, we said that at this point we now have a really dilute filtrate, and if we let that pass all the way through and out the body, it would be getting rid of lots and lots of water, as we might want to do when we are very well hydrated. In contrast, if we were able to reabsorb water at this stage, then that would allow us to produce a very concentrated urine. And because the collecting duct is traveling through the medulla that has the same concentration gradient over here as it did on the left of the diagram, I could get as concentrated as 1200 milliosmolar in the filtrate that is going to become the urine. And just a note on terminology, excretion means the removal from the body. So the filtrate here is going to be excreted as urine. Excretion is different from secretion. Secretion is transporting things from the blood into the tubule, and excretion is getting rid of stuff from the body altogether. So just to come back to the collecting duct here, water can be reabsorbed along the way to make a urine that is more concentrated, but that's actually controlled, as we talked about. We might want to make a concentrated urine sometimes and a dilute urine at other times, and that's controlled by a hormone called antidiuretic hormone, or ADH, and in the presence of ADH, aquaporins, which are transporters for water, will be put into the collecting duct membrane and allow water reabsorption. In the absence of ADH, there will be no aquaporins, and the dilute filtrate will continue straight through. We'll take a look at that process more in the next video lesson so that we can understand it in more detail. But to finish up here, although we said about 180 liters a day of fluid passes into the tubular elements of all of your nephrons altogether, by the end, because so much has been reabsorbed, you only produce about 1 or 1.5 liters of urine per day. So that's a look at the overall function and physiology of the nephron, and in the next lesson we'll look in more detail at how ADH controls the permeability of the collecting duct.